Welcome back, everybody. Hope you had great holidays. I just wanted to update you on some upcoming Grand Rounds presentations, and which are on the 11th. Joy Hirsch from our department will be presenting on her work where she uses a technique called near-infrared spectroscopy, which enables one to measure brain activity without going to an MR center. And, but she wires up two people at the same time and looks at their brain activity while they're doing social behavior, so the coordinated activity of their brains in social activities, which is really neat and very novel, and she'll be talking about that. And then the following week, we have Holly Lisenby from NIMH. She's the director of the Division of oh, I'm sorry, I think I just advanced your slides by accident. She's the director of translational research at NIMH, and she is also an expert in brain stimulation research, and she'll be talking about brain stimulation research. And I'm in particular hoping that she'll talk about a technique that she has been involved in developing, which is involved an alternative to electroconvulsive therapy where the seizure is precipitated by a magnetic stimulation rather than an electrical stimulation. It's called magnetic seizure therapy, and it has the possibility of being just as effective as electroconvulsive therapy, but a lot safer in terms of reduced memory risk. So that should be a lot of fun. And also, you may have heard that the dean has announced that he's not going to stand for reappointment. And so I don't know anything more than that, but certainly probably over the next couple of years, we'll have a new dean in the department. With all due deliberate speed, I believe a search committee will be assembled. We've heard that the provost and president intend for there to be a very full search, not a quick turnaround. And Dean Alpern has agreed to stay on as dean until the search is completed, so that makes it possible to have a full and deliberate search without pressure, so time pressure. So I'll let you know more when I hear more. And if you have any good ideas about people you'd like to be dean, you can send them to me or to the president, President Salovey, and you know, it would be good to have a good dean coming up. So, without further ado, let me introduce Marina, Dr. Marina Picciotto, who is going to introduce Alex Kwan, our own Alex Kwan, who's giving grand rounds today.
Welcome, everybody. I am excited to introduce Alex. So Alex has come to us from a journey from those esoteric realms of physics, where he was a physics undergrad, to the applied physics realm, where he did his PhD at Cornell with Watt Webb, looking at processes that are involved in the kinds of tools he uses now in terms of visualizing things that are really hard to see. And then Alex was lured to the dark side of neuroscience, and as a postdoc at Berkeley, he started to do the kinds of really interesting in vivo real-time imaging that you'll hear about now. So what he is able to do is to take mice who are behaving, doing the kinds of tasks that we hope are indicative of either primary behaviors that the brain initiates, or behaviors that are many ways relevant to psychiatric illness that we're interested in this department, and to analyze the large-scale circuit-level and network-level activity that underlies that. And why that's important is that we can look at single-cell events in the mouse. It's very hard to take that to the whole human brain. We need many levels in between that single neuron, the groups of neurons, the circuit, the overall brain activity that perhaps we can measure with functional magnetic resonance imaging or other techniques in the human. And to be able to put all of that together is part of our translational mission as a department. And so what's really exceptionally encouraging about Alex is that going from this physics background through all of these levels to neuroscience, he's made the next step and become a translational neuroscientist in our department, and has collaborated not only with other neuroscientists in the department, but also with people who are doing human lab studies, human imaging studies, and to those who are modeling the kinds of processes that are important for the dysfunction in psychiatric illness, but also how therapeutics may, in fact, intervene to change the brain to try to get these kinds of circuits back into a normal range, or I don't want to say normal, back into a functional state. So what I hope you'll hear from him today is an accessible presentation for all of us on how he uses this extremely sophisticated set of tools in awake and behaving mice, how this is relevant to human psychiatric illness, and how this may, in fact, provide insights for the kinds of new treatments like ketamine, or for really complex psychiatric illness such as schizophrenia in humans. So, Alex?
Hello, I hope you can hear me. Well, thank you, Marina, for the kind introduction. So I thought I would actually start by giving you a broader sense on what my lab does before going into some specific projects. So at Yale, my lab, we are mainly interested in two topics, kind of half and half. The first topic that we're very interested in is the role of frontal cortex in decision making. And in this set of projects, we're particularly interested in environments where the environment is uncertain or is dynamic. We're very interested in adaptive behavior, where animals have to be flexible. And in that sense, we're very interested in how animals learn different sensory motor associations, as well as we have some more recent interest in value-based decisions. So we have published a couple of papers in these areas in the last several years. I won't talk too much about that. I'll be happy to talk to any of you at a later time about that work. But instead, what I'll focus on is another half of my lab where we're actually interested in using some of the very similar optical tool and techniques in the mouse, but look at dendrites and dendritic spines in the context of psychiatric conditions. And some of the methods that I'll highlight in the talk and that we heavily use in the lab is optical methods, many of which carry, as Marine suggested from my PhD work, using some of these nonlinear microscopy techniques to try to image as well as perturb neuroactivity. We work with mouse, and increasingly, at least for the decision-making part of the program, we're developing some computational models for the behavior. So I thought I'd just start by being very broad, just first introducing dendrites. Dendrites is definitely a primary compartment of neurons, and I would venture to say that is definitely a unique aspect of neuron in that among the different cell types, in that you have a cell body that emanates from it a very clear, in this case, it's layer 5 pyramidal neurons in the cortex, emanates a very distinct and thick apical trunk. And then when it reaches the surface of the brain, it fans out. And you can see on the dendrites, they're on the orders of thousands to 10,000s of these little protrusions that are dendritic spines. Mostly dendritic spines contains functional synaptic contacts. So within the dendritic spines, on the scale of several micron, they have machineries for mediating synaptic transmission. So these are sites of excitatory transmission. They're glutamate receptors. And then another fascinating aspect is that the compartments are very isolated, right? So both biochemical signaling as well as electrical signaling can be confined within spine as well as spread across the dendrites. And another central feature of them is structural and functional plasticity. So not only are they important for transmission, but they can also change over time, both in terms of structurally, in terms of forming new spines, pruning old spines, as well as functionally. So even if you have the spine, the synaptic strength can change over time. And as a result, I think it's somewhat widely agreed that these are probably very essential building blocks for learning and memory in the brain. But another key aspect for dendritic spine is that dendrites and dendritic spines are also known to be very vulnerable for neuropsychiatric disorders. In fact, I would venture to say probably all disorders have some type of spine deficit. I don't know of really one where this has not been implicated. I like this review from Bonnie Feinstein, where they just have a cartoon showing the various type of changes that you see across different types of disorder. Here we have a normal neurons with the arborization coming down and spines along a branch. Of particular interest to us, you can see schizophrenia. There's a lot of work from labs like from David Lewis' lab showing that you have impoverished arborization as well as fewer dendritic spines in postmortem human tissues in the dorsolateral prefrontal cortex. But as well, for example, if you have stress and anxiety in major depressive disorder, you also see fewer dendritic spines. But that's not always the case, right? Some other disorder, for example, autism or Fragile X, it might have actually more spines or more phenotypia. And this is only structurally. Functionally, there's also perturbations. So it's really a central feature of psychiatric conditions. And I think that the idea that is central to the disorder is important for a number of reasons. The most exciting to me is that if the spines and more broadly synapses are dysfunctional, then that suggests that a good way to normalize or at least make a functional correction for this disorder might be to actually harness the inherent synaptic plasticity that's in the brain. And I mean, this is, I think, a view that's widely shared by the community, but also particularly in this department. Here's a couple of reviews by Ron and Jerry and John, suggesting that it might be very useful to harness that synaptic plasticity. So it's very important to understand the role of dendritic spines in some of these conditions. I think one of the challenges, though, is that when you look at a picture like this, though, a lot of what we know is static. So here's a static snapshot, another snapshot. We know that going from the more of a normal conditions to depressed conditions, perhaps you see that the spine has shrunk, but we don't know what's going on in the middle, right? We have an arrow here, and it's somewhat graphical, but we don't know how it goes, very little about how it goes from here to here, or even some of the signaling. So here you have an NMDA receptor, AMPA receptor, there's a signaling. We don't know as much about the dynamics of these type of signaling. So that's a gap that we're trying to fill. And just to really try to pound that message a little bit more, most of what we know are from static snapshot, either postmortem tissue or from reduced preparation like cell culture or animals that we have sacrificed. But instead, a lot about dendritic spines and synapses are quite dynamic, one of which is synaptic architecture. We know that spines can form, and then also spine can be eliminated. So here, I show you just a schematic here. Most of the spine probably stable in the brain. Sometimes you have phenotypia that are extension that could become stable spines, although some of them can be also transient. Some spines are eliminated, some spines are formed. And these are very important not only for learning and memory, but also for neuropsychiatric development. The spine number really changes quite dramatically over development, and many of these has been implicated in disorders as well. Not only the architecture dynamic, but the signaling is also tremendously dynamic. These are a lot of the molecules that's been studied at the postsynaptic density. But if you look at how they're actually elevated in terms of their concentration in the postsynaptic density in the spine, you see that there's a time scale to it. When the spine is activated, there's a calcium influx on the order of seconds, and then it activates a cascade of molecular pathway that also have a particular spatial temporal dynamics, some of which is spine-specific, some of which actually spread to the dendritic branches, some of which is on the order of seconds, some of it up to minutes and maybe hours, eventually lead to underlie the structural plasticity. So overall, I would argue that both structure and functionally, spines are extremely dynamic, and it will be very important to understand this dynamics. So the tools in my lab to try to address some of these questions and measure the dynamics to answer some psychiatric questions is using in vivo optical imaging. Here are one slide explaining our kind of basic broad approach. What we do is we take a mouse, be it a normal mouse or a mouse that is transgenic. We take out a piece of the skull. This is typically in the medial frontal cortex, so very close to the midline, about 2 to 3 millimeter diameter. And then you can see that once you replace it with a glass window, the skull, you can see the underlying microvasculature becomes optically transparent. We put it under a 2-photon microscope, which is a laser scanning optical microscopy technique, which allows us then to image a field of view within this 2 millimeter diameter, which is actually just this tiny square, which is projected here. So you can see that with this kind of imaging techniques, we have subcellular resolution. You can see, so this is an image as we try to take in different planes as we go into the brain. You can see the dendritic branches that come out of these layer 5 pyramidal neurons. And if we zoom in, you can actually then see the individual dendritic spines along these branches. So we have both now the spatial resolution to look at spines and branches, as well as because the optical window is chronic, we can now also track these things in order of seconds within a session, but also across days. So using these kind of approaches, what we're trying to get at, again, is trying to look at dendritic dynamics and try to answer some psychiatric-related questions. So in this talk, I will talk about 2 very specific projects, 2 questions that we have that we thought that this type of tool would be very important that we can address. The first question that I will ask, project we'll talk about, is about dendritic architecture, structure of the dendritic operization. And the question that we have was, if we give a single dose of ketamine, what is the dynamic longitudinal effect on the dendritic architecture? And this was work led by a great technician in the lab, Victoria, who was here about a year ago. So I show a slide that probably everybody know here, but this is the effect of ketamine on healthy humans. So it's quite widely known now, I think, and been replicated many times, that if you give human or mice a single dose of ketamine at a subanesthetic range, what happens acutely within an hour and then end at about 2 hours is that you have a range of psychotomimetic, perceptual, and cognitive symptoms. And in mice, it's hard to measure these, but you also get hyperlocomotion and other types of symptoms that might resemble some of these psychotomimetic effects. But it's also known that once this acute effect has largely subsided, there's also a rapid onset antidepressant effect. So I overlay some of the plot here. You can see that after it subsides, you can start to see a sustained antidepressant effect. It's rapid relative to other forms of antidepressants that is conventionally known, and it's sustained here, at least in the first study. They show up to 3 days, but subsequently, it can maybe last up to even 1 or 2 weeks. So this has been quite interesting, and I think there's a lot of work in trying to understand why that is and what might be some of the neurobiological underpinning that might underlie this antidepressant effect. And one of the, I think, strong lead is that it's actually also work done by this department by people like Ron and George Akaginian, where they show, building on some of the older studies that show that if you have chronic stress applied to rodents, you see a loss of arborization and also a fewer dendritic spines, that perhaps what ketamine does is actually to counteract that and bring it back, so it might actually allow the dendritic branching to be replenished, or if not that, at least you have a higher density of dendritic spines. So there might be a structural basis for the ketamine's action. But there still remains some question. Again, I want to stress that these processes are dynamic. So the question that we want to ask in this context is, okay, we know that probably ketamine increases the number of spines. Can we say something about how this increases spine arises? Is it because there are more spine that has been formed, or is it because there are less spine that's been eliminated, or is it because of both? What is the turnover dynamics that actually lead to the higher spine density? And if we can try to start looking at some of these turnover dynamics, then we can maybe also ask, what is the time course? And how does that time course, in terms of the structural plasticity, relate to the time course that we have seen for the efficacy of this drug. So to answer the question, we used in vivo optical imaging again. So we use this Phi1 GFP-M mouse where layer 5 neurons in the cortex expresses a green fluorescent protein, so we can image the arborization coming out from these neurons. Here is one image that we get, a full field view that we get. You can see the arborization, and we zoom in, then you can see the individual dendritic spines. So I note that we're imaging more the superficial layer in these areas. So we're not really imaging the cell body, but these are the tufts that comes out on the top that fans out around layer 1. So we have a very simple paradigm where we decided to do longitudinal imaging with the chronic optical window. We image twice before injection, and the injection is either a single dose of ketamine or saline, ketamine at 10 mg per k IP into the mouse. We don't image it during that time to not interfere with the drug's action, and then we image 5 more times subsequent to that single dose of injection. So then we have both the pre-injection period as well as the post-injection up to about 2 weeks. And to Victoria's credit, we analyze over thousands spines in both conditions. So this is the kind of data we can get out of this method. Here is a dendritic branch that we can track over a number of sessions. Here is the first time we look at this branch, day -3, -1. You can see there's a stable spine indicated by the green arrow that's actually stable all the way up until day 15, the last time that we look at this area. You can also see some changes. I mean, here's another stable spine, but you can also see some changes where initially there's nothing going on, but on day 10, you start to see that there's another spine that formed next to the stable spine. So this kind of technique allows us to have a record on where the spines form and where they get eliminated. And one of the first things that we notice is that when you compare ketamine and saline injection, we do confirm and validate some of the older results that has been shown, where we see that relative to saline, in our case, ketamine actually has a relatively higher spine density compared to saline. So if you inject a mouse with ketamine, you can see that the spine density change is quite a bit more elevated than control. One of the interesting things is you can see the control actually decreases quite a lot. I mean, you would expect that maybe if you inject saline, it should not do much to the animal. I think there are 2 more factors here, one being that some of these animals are younger. They're in adolescence, so it's probably a time when there's actually some natural pruning going on. Another thing is that the mouse, the imaging itself is still somewhat of a stressful event, I think. You have to take the mouse every day and try to image it. It's probably not very pleasant to the animal, so there's maybe some baseline stress level. But regardless, I think the design allows us to directly compare the injection for ketamine and saline. And the elevated spine density matches quite well with what has been reported by other labs at fixed time points. So the question that we have that I initially posed is, okay, we see more spine. Is it because of formation or elimination? And I think the data is quite clear here now, showing that it's a spine formation, an elevated spine formation rate that's driving this change. If we look at whether spine has been added or spine has been subtracted, we see that right after the injection of ketamine, there's a very notable increase in the spine formation rate. And this formation rate is sustained and above 0 all the way up to about day 10. In comparison, you can look at the elimination rate as well as some of the turnover dynamics on the saline case, and there's really not that much difference from 0. So most of the changes that we see, I think, in terms of the difference between the ketamine and the saline can be attributed to a elevated spine formation rate. So more new spines are being formed. One of the questions that we have, and it's not also, I think, harder to address in a static point of view is, okay, there are more spines, but are these spines actually useful? Are they actually synapses? Because there could be photophobia, spines could be transient. And it's been previously shown that if you do, this is one of the best studies I've been showing this, do a correlated optical imaging as well as electron microscopy study to show that some of these spines actually don't have any kind of postsynaptic machinery. This is a spine that's 0 day old, it's a brand new spine. But once you start looking at 4+ days, more than 4 days, here's a 5-day spine, 5-day-old spine, 9-day-old spine, then you start to see that they have features of synapses at the EM level. So we then also ask, are some of these new spines also can persist over time? And the answer is yes, but not all of them. If you look at saline, a lot of the new spine that we see persists. Ketamine, there are more new spine that form, but on the whole, actually, that fraction of new spine that persists decreases over time significantly. So what that means is that ketamine leads to a increase of a lot of spine, but some of those are transient, and some of them are stable.
Alex, this is really interesting, and it raises a question that I've been struggling with for a long time, which is there some counter, does the dendrite not like to have so many spines as created by ketamine? And is there a counter-regulatory process that ketamine is evoking that's actually reducing the stability of spines that are made by ketamine and accounting for the transience of the antidepressant effects of ketamine? To me, this is really an important question, like why doesn't ketamine cure depression? And what is the fact that makes what is the process that makes the antidepressant effect go away?
Right.
And so is this a signature of that, do you think?
I mean, I think it could be. I think there's a good amount of evidence suggesting there's a lot of homeostatic process in the brain that regulate synaptic strength and spine numbers. And in fact, I'll show you some evidence later that we're also trying to argue this, that I'll give the punchline away, but that maybe ketamine does not simply add spine, but rather the action is more reorganizing the synaptic inputs. Yeah. So to that, I mean, we also find some other changes that were a little bit more unexpected related to ketamine. So the spine increase has been noted before. What we also seen, and just looking at images, we just saw this thing, is that there's also some dramatic loss in terms of the apical tuft branch tips. So we see spine increases, but you can also see a branch here where we can see in the very first imaging session, day -3, that we don't actually see on the day immediately after ketamine. You can see this whole branch is not here anymore. Everything else is there. And I just want to show that this is not because the brain has tilted or maybe we missed it. You can go 10 micron above, 10 micron below. You just scan up and down. It's just really not there. So it suggests that not only it adds spine, it can also have some other changes to the dendritic architecture that's maybe a little bit more dramatic. Although you have to keep in mind these are not the apical trunk, right? Probably the cell is not dead. It's just the very tip in the layer 1. It's still probably a minor fraction of the dendritic element. And this effect is actually extremely acute, unlike the spine formation rate changes. You can see here again another example of this, another example of the branch loss. I think I don't see the branch loss. Maybe this is showing it's quite stable. I think in other cases, I mean in general you don't expect your dendritic architecture to change like that. But what we see is that with ketamine at least you can see a very acute drop in the branch length. Very much aligned to the time of the injection of the ketamine, and it's not sustained. So actually, so here's the conclusion now for this first project, where what we saw is that we saw elevated spine density, and that we can attribute it to increased spine density formation. So suggesting that new connections are added gradually. Since we published this work, actually it's quite nice to see that there are 2 now independent studies that also show similar conclusions, although in a slightly different context. One of them actually working with stress animal, where they explicitly stress with restrained stress, and then another one in just a different area of the cortex, the somatosensory cortex, as opposed to the frontal cortex. Also showing, both of them showing that you can have an elevated spine formation rate in response to ketamine. Second thing that we show is that not only you have spine formation, you have a branch loss. So some of the old connections are removed immediately. So what we think is actually what John or Richard alluded to, which is that you gain some spine, you lose some connections, and we think, we're just hypothesizing, speculating that on the whole, the net result might not be simply addition, but rather reorganization of the synaptic connection. Chris, yeah.
Alex, if you look out to 10 and 15 days, do the branches come back?
They never come back here, we
So
see.
the spines increase but then decline, and the branches go away and then never come back. So the net effect at 15 days is fewer branches with the same number of spines, which might be net loss of synaptic connectivity, mightn't it?
Well, there's an increase in formation rate, so then
At 5 days.
and then
But it
at
goes
5
down at
days.
15.
Yeah, but once those
after ketamine wears off, if the branches don't come back,
right.
and the spines do decline, then your total number of synaptic connections might actually be lower at 15
Right.
days than it was at baseline.
Right, but the formation rate is a dynamic thing. So there are more spines that got formed, and then a good amount of those spines persist over time afterwards, even though your formation rate is not elevated later. So those added spine at the time when the formation rate is heightened just remains with the system.
So you may have more spines on fewer branches.
You might have more spine on fewer branches. Yes, 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 yes. Yeah, I try to emphasize it. Yeah, so I think in terms of the dendritic volume, should not be that much. Yeah, yeah. I mean, there's some fMRI study showing that ketamine at a higher dose, in human, you can have some also white matter loss. Yeah, yeah. So I mean, again, this is a model that we have. It's a speculation that we're trying to do more work to show now. But if you imagine one scenario is that you can have input to this area from a red brain region and some blue brain region with different axons. You have the spines receiving different kinds of input. And if you lose one branch that receive mostly the red input and you add some other spines, it's possible that you can actually shift the kinds of inputs into this brain area. So I mean, proving this kind of model would require us to look at spines with specific opposing axonal terminals. And that's something that we can try to do with some two-color imaging technique to try to see if these might be like thalamic inputs or rather cortical inputs or amygdala inputs. I think that would be very interesting. So I'll move on to the second part, which is a little bit longer actually, where we're now trying to look at, instead of structure, functional changes in the dendritic spines. And this has to do with dendritic calcium signaling. And the question there was, what is the impact of schizophrenia-related alterations on some of these calcium dynamics that we can see that I've alluded to in the dendritic spines? And this work is led by a very talented postdoc, Farhan Ali, in the lab. So to motivate this topic, I did a lot of work showing implicating dendritic spine deficit in schizophrenia. It's definitely a central feature of the pathology. I think two of the strongest evidence are here as follows. One of which is from David Lewis's group, where they look at postmortem tissues to show that spine density is reduced in schizophrenia patients. These are actually, in their case, I think, basal dendrites and layer 2-3 pyramidal neurons. It's not exactly clear how this directly translates to rodent. Rodent don't have a DLPFC. There's also some indication, though, that this probably also is present in the cingulate cortex. It might be a bit more widespread. And then the other thing that I think actually is a bit more broad and definitely applied to rodent is the synaptic protein changes. So these are, it's a large-scale analysis of de novo mutation, and they show that a lot of the mutation can be associated with postsynaptic proteins. You can note that also there's a lot of sharing going on between schizophrenia and some of the other disorders. But there are just literally tens and hundreds of these mutations that can be linked to the synapses in schizophrenia patients. So the question that we have is, what are some of these impacts on dendritic spines, but in an awake brain? Most of these are postmortem studies, genetic studies. What if you knock some of these alterations into an awake brain? So the approach that we take is, again, an optical one. We have mice. In this time, instead of having a static fluorophore, we inject it with a calcium sensor. So this is a calcium sensor called GCAM6, F for fast. We also use S for slow, different dynamics. We inject it into the medial frontal cortex of the mouse, which is the CG1, M2 area. Right here, you can see the neurons transfected with this fluorescent sensor. And what we're going to do is you have these neurons labeled. We're going to image the layer 1 where we can see the dendritic spines. And here's an image of the kind of dendritic branches in the spines that we can see in a live mouse. So how does it look like in an awake Heffix mouse? This is the kind of image that we get, the kind of movie that we can get. This is a whole field view of the brain, about 200 to 300 micron across. You can see that there's just a lot of activity, dendritic activity. At this scale, you can see mostly branch activity. I mean, these are tuft branches that probably belong to the different neurons. This is a awake mouse. It's actually not doing any tasks. It's just just resting. And there's just a lot of things going on, a lot of calcium influx into these dendritic branches. To try to understand it, I want to explain a little bit how calcium actually get into the dendrites. So we're mostly interested in dendritic spines, so I'm going to focus on signal that we record from this area. And calcium can get into the spine in two different ways, right? One is if the spine receives excitatory synaptic input, then through AMPA receptor, NMDA receptor, there's a depolarization, right, of the membrane potential, and that would lead to calcium influx through a range of processes, probably including NMDA receptor, but also some other voltage-gated calcium channels. So that's a localized influx of calcium right here in the spine. But there's also a different mechanism that calcium can go in, and that's through back-propagating action potential or dendritic spikes. So these are more widespread events where the whole dendritic arborization get depolarized. So it would depolarize, and then eventually the spine also get depolarized. And this leads to calcium influx in the shaft, but also into the spine. They're somewhat different, because here the influx is through NMDA receptors, so it requires glutamate. But here there's not really a synaptic input, it's just a depolarization, but calcium can also go in. So these are different events that calcium can go into the spines. So we're primarily interested in the excitatory synaptic input part. But just to show you how these look like in vivo and how we can distinguish them, we can actually zoom into a particular dendritic branch. You can see that this particular spine, I like it because it's very obvious. You can see that sometimes it flashes on its own, so suggesting localized calcium influx in response to synaptic input. Sometimes the whole branch flashes along with the spine, suggesting some kind of more widespread electrical activity. So, and then here is a static snapshot of that from that movie, showing again here a widespread event, whole branch is activated. Here's a more localized calcium event showing that calcium influx into the spines. So for the rest of this talk, I'm actually just going to focus on these localized calcium influx. We're going to regress out components like this. Whenever we see the whole thing light up, we're going to subtract it off. And then we're going to convert these events, DF/F changes into event rates. But the idea is that if we focus on this localized calcium influx, then it would reflect changes that are related to excitatory synaptic input rather than some of the other dendritic events. So more localized to the synaptic function. To show that this subtraction method is actually kosher and that we're actually capturing the synaptic event, because it's somewhat of a post hoc analytical method, we want to experimentally confirm it. And to show that the event that we record from the spines is actually synaptically in origin, we did two-color imaging. So we loaded a red calcium indicator into the dendrites and also a green indicator into the axons. And we can then look at what happens whenever we see an event at the spine, whether we also see a corresponding event in the axon. Presumably, if this is synaptically driven, we should also see a corresponding presynaptic event. So here I show you that indeed these things are pretty correlated. You look at the bouton and here the spine, you can already see by eye that the events are quite correlated. And if we look at the probability of this happening, we can find that about 65% of the event that we detect on the postsynaptic side of things in the spine can be attributed to has a corresponding presynaptic calcium event. So the majority of what I'll show you is, I think we can be quite confident to say that it's probably synaptically evoked. So we want to understand how these calcium influx and synaptic excitability is perturbed by schizophrenia link alteration. There's a lot of different kinds of genetic perturbation that has relationship to schizophrenia. We decided to pick one that's actually a rare de novo mutation, with the idea that as a rare mutation, it probably has a higher penetrance, and that in an animal model that might reproduce some of the pathophysiology a little bit better than common variants. This is one particular de novo mutation that was discovered 8 years ago. There was a parents that is unaffected, and they have three sons, brothers that all have symptoms related to schizophrenia. And the mutation is then linked back to a particular mutation in the protein SHANK3. And then subsequently, Guoping Feng's lab in MIT has generated a transgenic mouse, knocking in this mutation into a mouse. So we took this mouse and we did the dendritic imaging I just showed you. What we found was quite striking in terms of the dendritic calcium transient. We see a fairly dramatic increase in the amount of calcium transients in the spines in the medial frontal cortex. So here, the two particular spines that we look at, you can look at on the same branch. Here is the transient we observe. Here is taking a mutant mouse now, two adjacent spines. You can see even adjacent spine can sometimes have different kinds of calcium transient, but on the whole, you can see that there's a really massive elevation, both in terms of the rate as well as the size of the calcium transients in the single spines. And here's a quantification of about 500 spines in each type of animals. You can see that if we look at just the rate of these events, you can see that there's a rightward shift in the cumulative distribution, suggesting that there's just more of these events going on in the mutant mouse once you knock in the schizophrenia alteration. So I think if this is true and is really relevant for the pathophysiology, then we want to make sure that we can probably see it again in a different kinds of alteration. So as opposed to a genetic perturbation, we also try a pharmacological perturbation. As I already said before, the acute effects of ketamine on an animal there's also some resemblance to schizophrenia. So we decided to also look at the acute impact of ketamine on these mice. Now it's normal mice. So we first did some imaging before, and then we can also image after the injection of ketamine or saline, but now looking at the acute effects. What we found was largely the same. So we see that in ketamine also really elevates some of these amplitudes and rates of the calcium influx in the spines, which is not apparent in saline, and it's quite different pre-injection. And here we have a within-subject comparison. Before injection, after injection, you see a massive increase in the calcium rate. And it's also borne out by the population analysis. So now we have two different kinds of alteration. One is genetic, one is pharmacology, that seems to converge under the same local circuit phenotype. Try to take a step back to see why this might occur. I show you that there's an enhanced calcium influx into the spines, and this is probably mediated through some combination of NMDA receptor as well as voltage-gated calcium channels in the spines. But I mean, that's kind of a little bit not intuitive and I think surprising to us, because if you think about what these protein does and what the mutations do, SHANK3 is a postsynaptic scaffold protein, and that mutation is known to cause a partial loss of function. So if you actually lose some of these scaffolding and lose some of the NMDA signaling, the direct action on the spine, you actually expect probably a reduction in the calcium influx. Same thing for the ketamine, which is known to have a primary action, at least on NMDA receptor. If you block the receptor, the direct action on that spine should be to reduce calcium influx. So it seems like that probably could not be explained by direct action on the spine itself. So that's why we hypothesize that they're probably more of a circuit-level mechanism. And in particular, we suspect that perhaps some somatostatin neurons are involved. There's a number of studies showing that these neurons are inhibitory neurons, GABAergic neurons, that make synapses onto dendritic branches, as well as onto spines to apply inhibition to regulate excitability of the dendrite in the spines. So the idea is that if some of these proteins and drugs actually act on and changes the amount of inhibition, then that could at least tilt the balance in terms of their direct action on the spines, and perhaps lead to the elevated calcium influx. So to test this idea, we can actually go and directly measure the somatostatin neurons. So we have SST Cre driver lines, and then we can have Cre-dependent GCAM virus. This allows us to load the calcium indicators specifically into these specific cell type, and we do the same imaging in the mutant mice, and we find that now instead of a rightward shift, we have a leftward shift, suggesting that we have less calcium events. And here is just example cells showing that there's a pretty noted decrease in the amount of somatostatin neuron activity, suggesting that we have a decreased amount of dendritic inhibition. We also want to do this more causally, trying to really link that decreased activity to the changes in the spine dynamics that we saw. So we want to manipulate the somatostatin neurons. And one of the ways we decided to do that is to manipulate the GluN2B signaling in these neurons. And there are two reasons for that. One is that if you look at the single-cell transcriptomics, there's a really heavy dose, good dose of GluN2B in the somatostatin neuron relative to some of the other cell types, actually. Notably, there's also a lot of actually in the VIP cells. And also, if you look at adult animal in slices, you can also see that if you measure NMDA current, there's also a good amount of NMDA current even in adults. These are the low threshold spiking cells that relate to the SST cells. You can see that when you put the antagonist for the GluN2B, you can see a substantial drop in the current in these cell types, even in adults. So this is a receptor that's quite active in these cell types. So this will allow us to manipulate the excitability and NMDA signaling into these cell types. And this is what we would predict, right? So if this is true, that we have somatostatin neuron activity decrease, lead to a decrease in the dendritic inhibition, and that's why this becomes hyperexcitable and higher calcium transient, then we would expect if we can boost the amount of NMDA signaling here, this should increase some of these activity, and then as a result, perhaps restore the inhibition, or at least elevate it, and then try to correct some of these changes in the spine dynamics. So to do that, to boost the GluN2B signaling, we use lentivirus. So these are actually building on some of the constructs and validation with help from Ron Duman's lab. We have lentivirus that we can overexpress GluN2B. And here is a validation in cell culture showing that it's a tree-dependent manner to overexpress GluN2B. Because we don't know, so GluN2B is one part of the NMDA receptor, but there are also other parts. And GluN1 is also obligatory receptor. So we don't know which is the limiting factor in terms of making a functional NMDA receptor. We also actually overexpress GluN1. Here's a validation for that, lentivirus. So injecting both of these virus into the brain will allow us to elevate both of the GluN1 subunit as well as the GluN2B subunit, leading to more GluN2B, functional NMDA receptor with GluN2B in it. So when we do that, what happens? We see that, well, here's just a first validation. We inject 3 of these viruses all into the same area, put a window, and then try to image it. You can see that first it works, so that we have the tomato, which indicates the GluN1. We have the BFP, which is fused to this, the GluN2B. And then we have the GCAM6, which you're going to image the activity of. And here's a triple coloring imaging showing that there are indeed neurons that actually express all 3 components. When we enhance the GluN2B signaling in these somatostatin neuron, what we found is that the calcium event rate increases. In fact, it overshoots a little bit relative to the normal condition. I mean, we can't really control the amount of expression here. And it's definitely quite elevated relative to the mutant alone. So the first part borne out, we can boost the NMDA signaling in the somatostatin neuron and then increase activity. So the expectation is that it should also then push us to normalize some of the calcium dynamics we see in spines. And indeed, that's also what we found. We load the lentivirus into the somatostatin neuron, but now we image the spines. And what we found is that similarly, the dendritic spine calcium dynamics also then restore to levels that's more comparable to the control animals. So it seems like by restoring the dendritic inhibition, by boosting the somatostatin neuron, now we can correct the calcium dynamics deficit. So this provides some causal evidence that the somatostatin neurons and dendritic inhibition is indeed one part, at least, of that lead to the heightened synaptic calcium transient.
Sorry, that's in the shank mice?
Yeah, this is in the shank mice, yeah. So we crossed the SST Cree with the shank mice. These are still homozygous shank mice. This is also the shank mutant, yeah. One of the things that we want to look at is, okay, we have a local circuit deficit. We also want to see if there's actually some behavioral consequences for doing these things. One of the behaviors is known to involve the frontal cortex is trace sphere conditioning, which involves temporal association. The idea is quite simple. You present the animal with a cue, auditory cue, and then after a while, here 15 seconds, you give it a shock. If the frontal cortex is intact, then it should be able to bridge that temporal gap and then know that the stimulus lead to a shock, and then later on when you present the stimulus again, the animal should freeze. And then as a control, we also present CS minus, so stimulus that has not predicted for anything. So then when this stimulus come on, nothing should be happening. This is the result. So we loaded the lentivirus bilaterally now into the medial frontal cortex. This is the trace result. So There's a lot of bars that I'll go through slowly. Here's the normal mice, SST Cree mice. You can see that they do freeze for the trace field conditioning. The mutant, though, did not really freeze, suggesting that the frontal cortex is compromised. Now, if we put in the lentivirus, some of that freezing behavior is restored. So they can freeze again in the trace condition. Just to show that this is not a learning deficit quite specific, but it's really, actually, this trace is important. We also did delay field conditioning. Just with the normal mice as well as a mutant to show that the mutant is not, does not show a deficit in the delay field conditioning. So it does seem to be a very cortex-dependent manner. It's not just that the field conditioning itself is generally impaired. And the lentivirus has an effect on this behavioral phenotype. OK, so to summarize a little bit, I show you that in the mutant, if we boost the SST, specifically in SST cells, the GluN2B signaling, then I can raise the SST neuron activity, lower the spine calcium, and then also changes the frontal-dependent behavior. We tested 2 alterations. I think the problem with testing too many alterations is you have to replicate everything. So, so if you think about the ketamine now, then that would suggest that, so presumably it's act a different way, right? Ketamine probably then act on NMDA receptor here, reduces MTA signaling here, and therefore causes a lower activity, and then also in the same way lead to elevated spine dynamics. So what we would expect in a causal type of experiment would be that if we now knock down where ketamine actually, the target of ketamine in the SST cell, by knocking down this GluN2B or NMDA signaling, then we would expect to first, without even ketamine, knocking down this should reproduce ketamine's action. And furthermore, it should render additional ketamine ineffective. So to try this other way, instead of boosting signaling, well, I guess first we have to show that indeed in the ketamine case it's also reduced. So we did indeed see that. So here's a ketamine case. It's quite similar to the SHANK3. Pre-post, there's nothing going on. There's a myocenic neuron. Pre-post, you can see that after ketamine, so myocenic neuron activity also decreases. And then when we want to manipulate it, just as what I explained to you, the scheme is we should knock down GluN2B now. And this, again, worked with Ron. He has a very nice construct, AAV-based construct that can knock down GluN2B, and here's a validation. And I won't show you all the data, but trust me that we did everything, where when you knock down, it also reproduces ketamine action, decreases the myocenic neuron activity, elevates spine calcium, as well as render additional ketamine ineffective. So really, it can go both ways, bidirectional modulation of GluN2B, whether you boost it in the mutant or you can knock it down. In the case of ketamine, it both seem to provide some causal evidence, at least, that the spine deficit that we see is really inhibitory in nature. So here is what I just said, summarizing some of the results. So as one of the final bits for the second half, we want to go a bit further. So we have a local circuit phenotype with some behavioral consequences. But one of the things we're quite interested in is what does this mean for the broader circuit in terms of cortical networks? There's a lot of hints that the broader cortical network is altered in schizophrenia. So what we see is that we have elevated spine here, probably hyperexcitability. These spines become hyperexcitable. It's actually quite well known that these layer 1 spines in the frontal cortex receives a lot of long-range cortical inputs, synapses from other cortical regions. So we want to know and see if that itself is also perturbed. So to do that, we did some electrophysiology. We recorded LFP. So here I show you an LFP recording in this medial frontal area of the mouse where we did the imaging. This is a kind of a spectrogram that we get. So it's a frequency over time. One of the recent paper that I like a lot from this David Kleinfeld lab show that if you look at blood vessel dilation as well as record LFP, and then you from the LFP you look at this gamma band and you integrate the signal in this gamma band. This integrated gamma actually reflects quite well and correlates quite well with the blood vessel dilation. So it's a good proxy for both signals. So this is what we'll focus in. We recorded LFP and we'll record the integrated gamma band to look at long-range communication. This is what it looks like. Farhan recorded LFP from the medial frontal cortex as well as LFP from a more posterior retrospinal cortex. We want to look at how these areas are communicating on a kind of long-range cortical communication. Pre-ketamine, you can see that here's the traces. Post-ketamine, I think you can already see visually that post-ketamine, the signal becomes more correlated. So they becomes the functional connectivity, if you want to interpret these as similar to bowl, they're heightened. Actually, quite similar to some of the works from Allen and Tichevich showing that with ketamine, you have a hyper frontal connectivity. So to, yeah?
That's the time scale there. It's immediately after.
This is same thing, yeah. So we started doing these things about 30 minutes after the injection, yeah, and then up to about 60 minutes. So it's like acute effect of ketamine. So go back, try to tie everything together. We now want to manipulate the dendritic inhibition that we've shown have been so effective at changing the synaptic calcium input. Same LFP in both sites, but now we do this GluN2B knockdown into the frontal cortex. What we found is first that the knockdown itself, with the knockdown versus no knockdown, reproduces ketamine's effect, so it leads to a heightened functional connectivity between this area. Furthermore, if you look at with the knockdown, you can add ketamine, and ketamine is no longer effective in elevating this connectivity. So it seems that that synaptic deficit I showed you in layer 1 actually could have some consequences even on the broader cortical communication. So we're quite excited about this finding. So this is a summary of it now. We found that you lose dendritic inhibition, specifically through somatostatin expressing interneurons. These neurons target dendrites and spines, lead to synaptic calcium influx. And we're excited about this because, I mean, calcium influx into that area, as I showed you previously, lead to a cascade of molecular changes. It's extremely important as a regulator of synaptic plasticity, very important for neurocircuit development in terms of spine formation and maintenance of spines. Many of these things have been implicated in the pathophysiology, so we think it might be one of the very essential and key steps towards some of the maladaptive changes associated with the disorder, or at least some of these alterations, the effect of the alterations. And I've shown you also that there's abnormal long-range cortical connectivity. This is interesting, I think, because given a lot of work in the functional connectivity space, altered cortical network, this is actually somewhat of a cellular basis that could start to try to explain or try to understand some of the connectivity deficits. And I think it's quite interesting to think about this in terms of the more well-known disinhibition theory for the ketamine, for the NMDA receptor hypofunction. That's been around for, actually more than 10, maybe even 20 years before some of these reviews come out now, where there's been I think a very prevailing view in the field that not only disinhibition here, but you also have disinhibition in the parvalbumin cell, which I have not talked about here, perhaps acting in parallel or perhaps together. But disinhibition here leading not to hyperexcitable spines, but actually hyperexcitable cell bodies, leading to a range of deficits that's somewhat different, but also quite important: hyperactivity, excess glutamate, gamma band oscillations. So it'd be very interesting to see whether these type of disinhibition are, again, like, are they parallel? Do they contribute to different kinds of behavioral deficits, or do they act in concert? I mean, there's also a good, I think from basic science viewpoint, there's also a good indication that one important factor for pyramidal neurons is this kind of somatodendritic inhibitory axis, where you put the inhibition on the cell has a big impact on its operation. So the shift in these balance could be quite important. Okay, so in summary, and then 2 more slides after that. In summary, I've tried to motivate you that much of what we know about dendritic spines has been quite static from postmortem tissue and reduced preparation. But now we have an opportunity to capture the dynamics of spines over time and over a spatial scale using newer in vivo imaging techniques. I've shown you 2 results. One is that ketamine elevates spine formation rate, so more spines are forming, and this might lead to a reorganization of the synaptic inputs in that area. The other thing is that for 2 different types of schizophrenia link alteration, one genetic, one pharmacological, quite different alteration, they seem to converge into the same local circuit phenotype, namely the aberrant calcium influx into the dendritic spines. So what's next? So there are a couple of things that we're quite interested in trying to building on these studies. One of which is actually to connect the calcium influx back to the antidepressant effect. The ketamine lead to these acute effects, which we see, calcium influx. that then also have a structural impact later on down the road. So we want to really know whether these elevation that we thought might be very important for the structural plasticity are indeed an essential step toward that formation rate that we see later on. I mean, it seems to make a lot of sense because it's really the regulator in many of the biochemical signaling later on. But we should test that. And if it's true, then this kind of influx, which is, I would argue, is much easier to measure than counting the spines over many days, could be a very interesting biomarker to try to look for what the different drugs do. And that's, I think, one interesting point where this could be actually a point of convergence of different kind of drugs. You can imagine calcium influx influenced by ketamine through GABAergic neurons, but I mean, there are other things that you could also perturb. For example, voltage-gated calcium channels that could directly act on spine, that could also change the calcium influx, but maybe lead to a same local circuit phenotype, but with a different kind of receptor action. Another thing that we are interested in is chronic stress. So again, taking advantage of the longitudinal aspect of it, how we can go across time. We started doing a lot of social diffuse stress in the lab. Interesting thing about this behavior is that compared to control, you can see that some animals are not really, quite resilient to the stress. They don't really show any deficit in social interaction behavior. Some of them are quite vulnerable to the stress. And it would be very interesting to look at these dendritic changes, both in terms of structure as well as the function, pre, during, after stress, what happens across the time, whether some of these structural changes precede or come after some of the behavioral changes that we can see, and maybe some of the individual differences as well, because we can now track the same subject over time. Okay, so yeah, I want to thank my lab. So my lab has been quite instrumental in getting these work, in particular Farhan, who's really spearheaded the schizophrenia project, as well as Victoria, who did all the work for the ketamine and the spine formation. And I want to thank a collaborator as well as funding sources from NIMH and NIA. Thank you.